try to share some of the work I've been done in the past few months about how to stabilize the SARS-CoV-2 spike protein by closing it and make it the side effect of that is that it becomes sort of thermostable. And we hope that this technology will help development of better diagnostic reagent and or method, or lead to a better vaccine. So let me just start. Can you see the change of the slide?
Yes, it looks good.
Yeah, good. Okay, good. So I'm a structural biologist by training, actually. So I'm actually in the last 10 years or so, I've been focused on actually influenza virus spike protein with Young scale. But just before I joined the MRC LMB, I was working in David Weissler lab, actually in Washington, University of Washington in Seattle, and I was in that lab. I was working on spike protein of SARS-CoV-1, and I mean, there's a lot of parallel similarities between flu spike protein, which is hemagglutinin, and the SARS spike protein. And when I started work, people just about to start to reveal the structure of the coronavirus spike protein, because previously this protein could not be crystallized, and it was only when the cryo-EM started to kick off that people start to see the structure of this protein. And one of the first surprising things people found with this spike protein of coronavirus, in particular beta coronavirus, which SARS is in and SARS-CoV-2 is in, is that when they solved the structure back in 2017, that there are two conformations people can say by cryo-EM, which is the closed form in the left and the partly open form, one of the monomer within the spike protein trimer is open. So that's what people can say at the time. And there were actually, at the time, there were actually two studies saw similar effect, that where people found that one of the monomers within the spike protein trimer can open and close. At the time, it wasn't obvious that what's the effect of this structural dynamics. But when they compared with at the time, what was being solved is that it's certainly a very peculiar dynamics for the beta coronavirus, because, and in particular the MERS and SARS, because what they found was that previously the MHV, which is a beta coronavirus, and HKU1, which is also a beta coronavirus, as well as at the time that was the NO63, which is the alpha coronavirus. All these three coronavirus spike protein trimers, they only adopt this closed conformation. But in contrast, for the two beta coronavirus, the MERS and SARS, where they adopt this closed and open conformation. So at the time, it was quite puzzling. What does this thing do? And when I work on this protein, I was actually trying to look at how antibody recognize this protein. And then this is the structure I determined, which we solve the structure of antibody that's isolated from a SARS survivor. And when we solve the structure by cryo-EM, we found it's very surprising that the binding of this antibody actually opens three of the monomers of within a spike protein trimer. And they now adopt this very open conformation. And what we found was that the antibody, by opening it, it makes the spike protein very, very fragile. And by incubating it for a bit, for as little as half an hour, a lot of the spike protein turns into this post-fusion state, significanted by this kind of very thin rod structures. And if you incubate for a long period of time, all of these spike proteins convert into this post-fusion state. And they becomes this kind of rosette, because the fusion peptide is exposed. And we found similar effect with the receptor of spike protein ACE2. With ACE2, we found that it also has similar effect, but the effect is less pronounced because presumably the binding affinity of ACE2 to the spike protein receptor binding site, hence the ability to open up the trimer is less pronounced. And we then at that point figured out this open and close is a trigger for the spike protein to go into the post-diffusion state. And that and by working at it, we found that this this open state is very, very unstable, and that actually, so when we're working on this project, it was very difficult to obtain a complex, and we found that complex can only exist in a transient amount of time, and the protein we purify can only survive for a very short period of time because it automatically opens and closes. So when the SARS-CoV-2 started the epidemic in China, I was really worried about this effect because this self-close and open here is the structure that determined by David's lab. And then again, they saw this kind of closed and open state for the SARS-CoV-2 protein. And immediately I was actually really worried about this because I know that because of the coronavirus have this closed and open state, then the expressed protein can only be stored at most for about a week inside at that low temperature in the fridge. And it's a big contrast to the flu spike protein hemagglutinin I used to work on. The hemagglutinin can actually store at the prefusion state with for actually many years. Actually, if you do not expose the protein to low pH. And I thought that this property is really bad for making a successful vaccine because if the half-life of the spike protein, which is the major antigen for the SARS-CoV-2, if the half-life is really poor, how can you make a very good vaccine? And similar problems has been reported for HIV glycoprotein as well as for the RSV glycoprotein. So I thought it would be good that this problem can be solved, and that's why I started the project. So based on, and again, actually, at the time, shortly after the epidemic of the SARS-CoV-2, people also look at the purified SARS-CoV-2 variant, and after purification, so we now know that this can be a purification effect, but the purification effect is it's very standard purification effect. After going through the purification effect, people can say that on the SARS-CoV-2 variant, these very thin rod structures, and then by looking at very closely, they are post-fusion structure. And my prediction is that with this post-fusion structure on the virus variant, it's if you use this as a vaccine, similar to what people use for flu, then it's not going to work because this surface glycoprotein has gone into the post-fusion state, and it's not going to be the same as the native structure of what it is on the virus, and it's not going to be effective as a vaccine. So I'm really trying to solve this problem. So so shortly after we decided to do this, the structure of so we first tried to design this kind of stabilizing mutations based on SARS-CoV-1, but shortly after we started it, the SARS-CoV-2 structure was solved by two different labs, and then in this case we used the one from David's lab, a previous supervisor in Washington University, and we found that so we actually designed multiple different stabilizing mutants, but only the ones that here we shown here actually worked. And the idea for this particular mutant was that we could close the RBD domain to eliminate the structural dynamics. And to do that, we designed two disulfide crosslinks based on the structure, because these are two pairs of residues that are close enough to form a disulfide bond. The first pair, called X1, was between the serine 383 within the RBD and the 985 within the S2 region. And we also designed another pair, which is X2 between the 413 within the RBD and the 987 within the S2. And it should be noted that one of the residue is actually within this widely used stabilizing mutant, the double P actually. So the 987 and the 988 are double P are both polings in a very well known construct that's been designed by McNellan back in 2017. And one residue of that had to be mutated to form this disulfide bond. We also actually noticed that although that this widely used protein mutation can actually increase the yield for this protein, but it's not actually, it actually somehow not also destabilize the spike protein a little bit. So it should be quite careful when people use this this spike protein formula to as a basis for the vaccine. And I will introduce a bit further what we found for this specific stabilizing strategy, the protein stabilization. So so then we started the expression trial. So at the time we also so as I mentioned that the double P, we noticed that the double P actually in the previous SARS study that the double P in our hand is not particularly stable. So we also designed two sets of construct. One is that in the presence of double P, the stabilization mutations previously reported, that is significant, that is signified by this double P here. And we also designed another set, which is without the double P, basically that two polines are replaced by the native sequence, which is lysine and valine there. So we found that interestingly, for the X1 mutant, it's only expressed in significant amount in the double P background. So here is the Western blot of using the haystack of the protein. We can say that the expression of the spike protein monomer here, and we can say the higher oligomers at a higher molecular weight here. This is a long reducing blot. And then we also, the X2 mutant is only expressed in the wild type background without the double P. You can say that the X2 doesn't express in the presence of double P. And again, they also show this higher oligomeric state, and such in a way that there's not much of lower molecular monomer bond here. So it seems to be the cross-linking by the X2 within the wild type background is actually better than the X1 within the double P background. And then this is the reducing gel plot. You can say that pretty much all the oligomers for the X1 and the X2 are converted into monomers, meaning that the higher oligomers shown in the long reducing plot is because it's due to the disulfide bonding. So then we tried expressing this in the in sort of median scale level, about 500 ml of media, and then the spike protein was secreted into the media, and we can get about 200 micrograms of protein for 500 ml of XP293 cells, and this we expressed it. We expressed it X1 in the double P background. We expressed the X2 in the wild type background. Or in addition, we also engineered. We have this in two different variants. One of the variants is called GSAS, meaning that the polybasic site is replaced by the GSAS. We also have another variant of the polybasic site, which is the R, meaning that the polybasic site is reduced to a single arginine, similar to SARS, make it similar to the SARS-CoV-1 and its progenitor IATG13. We found that removing the polybasic site is crucial to get good yield for the spike protein. So as you can see from the gel, so here's the comparison. Normal spike protein expresses as when running on the STS page gel in either reducing or long reducing conditions is actually a monomer, with the X1 cross-linking disulfide linked mutant, we saw that like there's very little monomer, but we saw dimer and trimers here. For the X2, we can say that the protein like primarily exists in the trimeric state here, as shown here. And by reducing that, you can say that all these proteins converted into the monomeric state, and that means the protein the protein is disulfide linked as a trimer when under long reducing condition. And on this side, these are the similar expressions for the polybasis side reduced to the arginine, a single arginine variant of the protein. You can say similarly for the double P, for the X1 mutant in the double P background, that it exists as monomer, dimer, and trimer. And for the X2, it exists primarily in the trimeric state. And due to the, and by reducing gel, these all go to monomers. And because due to this, this observation, we actually focus mainly on the following serological studies based on the X2 protein variants. But before we do that, we want to look at the, on the serological study, we want to look at the physical characteristics of these different spike protein variants. So here is a lective stain image, the 6EN image of this long cross-linked S protein, long disulfide linked S protein. So this is the previously very widely reported construct, the spike protein with the double P stabilization mutation. So when it's freshly purified, the protein looks lovely with this kind of trimeric spikes everywhere. But if you store it at 4 degrees within a day, you can say that a lot of these trimeric spikes disappeared, and you see this kind of little dots, which is the disintegrated spike protein. And within 3 days, most of the trimeric spike has disappeared and then becomes very disordered kind of small dots. So the stability of these kind of spike proteins are very bad without either it's been stabilized by the double P or not. So because here is a sort of wild type kind of spike protein without the double P, but with the KV there, but the multibasis side is changed to arginine. You can say that it's very similar, but somehow it's sort of slightly more stable than the double P variants when stored at 4 degrees. And again, this is another one. The polybasis side changed to the GSAS, as reported by McLellan. Similarly, within 3 days, or most of the spike protein trimers are disintegrated. And again, if you also treat it at 60 degrees for about 30 minutes, you can also say that this kind of lovely, well-formed spike trimers becomes this all smaller species, presumably it's denatured. In contrast, if you look at our cross-linked bisulfide proteins, spike protein trimers, can say that we freshly purified, very nice, and without much of disintegration at all. And if we store at 4 degrees for either 3 days or even a month, that the protein pretty much stayed as a pre-fusion trimer, as signified by this kind of triangular kind of spike protein trimers. And our protein can also be heated up for 30 minutes at 60 degrees. And that still looks fine by lective stain EM. So because we are structural biologists, we want to have a further look at, is there anything special about this kind of stabilized protein? To do that, we used cryo-EM, and this is how we look at it. So we solved actually structured 4 different proteins. We have the wild type like spike. The only difference between the wild type, between the native sequence at the multi-basic sites now changed to a single arginine, so that is similar to SARS-CoV-1. And we solved this structure by taking the image, and then we did 2D classification, 3D classification, and we found that, so one of the things we found for the first few coronavirus spike protein papers, cryo-EM studies, is that we have very different close to open ratio. In those previously determined structures where they use the double P construct, a lot of the spike proteins are open, but in contrast, for the wild type without the double P, but replaced with the native sequence, KV, we saw that a lot of the spike proteins are closed. We saw 80% of spike protein are closed, while only 20% are open. So when we introduced the double P and we did the structure in a similar way, we found that in this case, with the double P, that the spike protein is actually more open. We got actually 80% of the spike protein are open, while only 20% of the spike protein are closed. And when we actually did a very careful classification, we also found that the closed conformation actually contains two further very subtle two conformations, very subtle differences. One is actually closed and one is locked, and we're going to talk about this in great detail in later on. So when we look at our stabilized protein by cross-linking with disulfide bond by cryo-EM, we found for the X2, which is very well, it only has trimers by SDS page. We found that all the spike proteins actually only have one single conformation, which is the closed conformation, which is the same as the wild type spike closed conformation. And for the X1, which is not fully cross-linked, we saw monomer, dimer, and trimer on the SDS page. We can say that all the proteins are closed, but within the closed population, 80% are closed, similar to the wild type here, as shown here. But there's a subpopulation, about 20% is in this locked conformation, which I'm going to talk in detail about that later. So, with the cryo-EM here on the left are the cryo-EM densities of the proteins we made. We can say that for these structures, the X2, X1 closed, and X1 locked, they are all closed trimers, and this is the top view with 3 different subunits in 3 different colors. And it's the side view here. The box indicate where the engineered disulfide bond are. So we can then look into that, and we saw that for the X2, that the engineered disulfide bond is definitely showing this density of disulfide bond here. And then we can say that from the structure, within the structure, the K986 is actually very close to the RBD residue 428 and 427. In the double P, the widely used stabilizing mutation that has been previously published is that this lysine residue is replaced by a proline, such that this electrostatic interaction is abolished. And that actually, and by our cryo-EM study, we found that by doing that, this makes the spike protein much more open. How would this effect affect the antigenicity of the spike protein used made with this double P mutation? It's not quite known, and then I think requires further study. And then we can say that for the X1 X1 cross-linking, we can say the disulfide bond here that has been engineered. Again, this shows that, and we hypothesize that we know that the X2 that cross-link much better. That is because the presence of the lysine here, and such that, as you can see earlier, that with the wild type sequence, the spike protein closes much more, and then that's probably why the X2 cross-link mutant actually is much more efficient in forming a fully cross-linked trimer. And here is trying to we're trying to look into what's the difference between the locked conformation and the so-called locked conformation and closed conformation. As I mentioned, we saw for the closed spike protein, we observed 2 distinct conformations. So you can say that this is alignment on the left, or the locked conformations aligned together from the the SR-P and SR-PX1 cross-link mutant. You can see that all these log conformations show very similar structures. For all these other structures we determine have this closed conformation, you can see that all these closed conformation are also very similar with some movement of the NTD. But when you overlay the locked and closed conformation together, you can see that for the locked conformation, which is this purple one, the RBD on the top, the S1 on the top is somehow rotated a little bit, such that the central channel there is becomes smaller by about 1.5 angstrom. Sounds like a very small effect, but then when we look at in detail, we found that in this lock conformation, we saw this refolding of this what between residue 833 to 855 residue, forming this yellow motif here. Here is the electron density. This is the cryo-ion density for that motif. You can see that this motif is indeed very well formed. And this motif is very interesting that it's upstream of the S2 prime site and the fusion peptide, as shown here. That is just above the, it's just N-terminal to the fusion peptide site. And then what the effect was that if you dock an open RBD in this locked structure, you can see that the open RBD actually would clash into this 833, 855 motif in the way that that means that the formation of the 833, 855 motif actually locks the spike protein RBD in the closed form. And that's why we call it a locked conformation, meaning that with the presence of 833 to 855 motif, the RBD is locked in the closed form. And this was actually very novel finding, because previously the SARS-CoV people did not observe this motif in SARS-CoV-1, and the first two SARS-CoV-2 spike protein paper, people didn't observe this motif. So I did the comparison with other coronavirus spike proteins where it was a mystery that why that people only observe for this coronavirus spike protein that there's only closed conformation for this spike protein. So meaning that was previously mentioned, the mouse hepatitis virus spike protein, that's an overlay, that's the NO63 spike protein, the MHV was in the beta coronavirus, the NO63 in the alpha, the IBV infectious bronchitis virus, that's in the gamma subgenus, and the post and delta coronavirus, which is in the delta family. And the surprising finding was that for these spike proteins of 4 different families, alpha, beta, gamma, and delta, for which the only closed form was observed by cryo-EM, such that in this one, by this comparison, all of these spike proteins have this equivalent 833 to 855 motif. And that actually solved the mystery that why these distinct spike proteins have only closed forms. It looks like this 833, 855 motif is actually a switch for the spike protein RBD to open and close.
Xiaoli, we have a couple minutes. We should wrap up.
Okay. So that's basically shows that. And we also show that this motif also has interaction with the D614G that is widely distributed now, and that controls the, and we hypothesize that the D614G mutation might affect the dynamics of this protein. And then we did focus on the X2 protein, which is homogeneous in a trimer. We did very serological assays. We showed that the X2 protein, this is the ELISA assay, with different multiple patient serum. We showed that the reactivity to the patient serum for the X2 protein is slightly less than the double P, which is widely used, and it's about 3-fold less. Here's basically a comparison of double P and X2 extracted from this 9, and this is the quantitative analysis. So it's about 2.5-fold decrease for the closed protein. That's probably because it's closed. And here's the Luminex assay with an tested various different construct, the double P, two different double P, the wild type ones that are very similar. For the X2, similarly, there's a slight decrease, but significantly, this closed protein displays significantly more epitopes than the RBD. And here's on the right is to show that while for the double P protein, the temperature have an effect on its antigenicity. At 60 degrees, the reactivity to the serum has significantly decreased. For the X2 protein, it's thermostable at 37 and 60 degrees. At the moment, actually, there's quite a lot of other studies that are now published together with this particular one. So there's one in NSMB and one still in the bioarchive. There's also a science one that has been published. And these now say all these reports has an aim to stabilize it. You can refer to for further detail. And what's the significance of this? So we found, I mean, there's now multiple reports that neutralization antibodies that can, or antibodies can actually disintegrate the spike. So we hope that by engineering this stabilized protein, such effect, it will maintain the antigenicity of the spike protein further without the effect of the serum, and that might help to make a better vaccine. And it's been reported that the RBD binding antibodies could also mimic the receptor with the assay interactions there. It may lead to the antibody-dependent enhancement for cell infection. And because the spike protein in the closed state, less antibodies may target to the RBD, and that might solve this problem. So here's the acknowledgement. The funding is from the MRC and ERC, and this is a very like large collaboration effort. And the initial gene was sent from China, from Guangdong. Shortly after the pandemic, the epidemic in China, like our colleague Andrew Carter, who is a PI in the institute, actually did the mutagenesis, and the student in the lab helped with protein production. While EM was done by Kun, Junlong, and Shores, very professionally. And the serum was done by these people in Royal Papaworth Hospital. They were collected and processed then. And the ELISA was done by Ian Goodfellow's group in Department of Virology. The Luminex assay with patient serums done in the Addenbrooke Hospital. They also used the same method to diagnose with our protein provided to diagnose a lot of patients for the hospital. And there's and James Leyson's group also provided some other crucial region. And finally, thank you for your time to listen to this.
Great. Xiaoli, thank you very much for that. Beautiful structures.
Yeah. Yeah.
So I'm curious about one thing. You mentioned that the virion prep showed much of the spike being in the postfusion state.
Yeah.
Which you would think would be not infectious anymore. And yet we're used to thinking of the virions as being relatively stable. And, you know, I wonder how that works.
So, I mean, this one, goes through this sucrose gradient, various different things, and ultracentrifugation. So I guess that's why it's not so stable. And then, as far as I know, some of the split variant vaccine that people trying to make need to go through similar process. So yeah, that could be the difference. But from my understanding, that in the environment, SARS-CoV-2, they are stable on the surface for probably about a day or so, something. So I thought
a day or two.
That's probably not the most. I don't know. Like that's probably not the most stable thing, I guess. Yeah. Comparing to, I don't know, for some other things.
Well, and I guess the other key, I mean, these stabilized forms are obviously crucially important for structure determination and so on, but I guess we don't really yet know in their context as a vaccine, you know, how it will go. Yeah.
So we actually did some preliminary immunization studies on mice, and we found that the X2 is the best. I mean, is much better than the previously published double P variants. But whether why that is, yeah, that's, I think, requires further investigation, because as I mentioned, you know, when initially people thought that the spike protein are a lot of them are open, but it may not be the case, basically. So probably a lot of the spike protein on the virion. So we also look at some virion without purification. We found most of the spike actually closed. So by stabilizing the spike protein in the closed form, and that's actually a more appropriate antigen for immunization. And in addition, I guess, because the closed form looks like it's much more stable, so the half-life within the serum may be more, maybe longer, and then that might help a better immune response. So I think it's definitely worth trying for this, you know, at least for that,
right.
to test this.
And whatever, I mean, whatever the platform is, all of these mutations will be, you know, readily usable in the sense that they would simply be made part of the
yes, hopefully that is, yeah. But I think some of the crosslink, the disulfide mutant, they may have, they may require a lower temperature, but it's not been thoroughly tested. So I observed definitely it expressed better at 33 degrees, 34 degrees, better than 37 degrees for some of the disulfide mutants. So that might be a problem, but yeah.